Now it's the turn of a second speaker, Professor Daniele Faccio. Daniele Faccio is a Royal Academy Chair in Emerging Technologies, Fellow of the Royal Society of Edinburgh, and Knight of the Order of the Star of Italy. He joined the University of Glasgow in 2017 as Professor in Quantum Technologies, where he leads the Extreme Light Group and is Director of Research for the School of Physics and Astronomy. He is also Adjunct Professor at the University of Arizona in Tucson, USA, and Fellow of the Optical Society of America. Previously, he was at Heriot-Watt University and University of Insubria in Italy, and he has also been visiting scientist at MIT, at ICFO in Barcelona, Spain, and ERC Fellow between 2012 and 2017. He was also awarded the Philip Leverhulme Prize in Physics in 2015, and the Royal Society of Edinburgh Senior Public Engagement Medal, and the Royal Society Wolfson Merit Award in 2017. His research is focused on the physics of light, in particular on how we harness light to answer fundamental questions and improve society. The title of the talk is Hong-Ou-Mandel Microscopy and Fluorescence Lifetime Imaging. Thank you again, Professor Daniele Faccio, for being here today, and please, the stage is yours.
Thank you. Thank you for your kind introduction, and thank you for everyone coming today. So thanks to Giuseppe for giving that really nice introduction and overview into sort of the basics of quantum imaging. So what I'll do is I won't repeat that. I'll dive straight in the deep end and start talking straight away about Hong-Ou-Mandel interference, which basically is an interference effect between indistinguishable photons. These photons can be entangled, but they don't have to be. What they have to be is indistinguishable at a quantum level, which means that in all their degrees of freedom, they have to be identical. When that happens, so what I'm outlining here in the top left is just the basic outline of a very simple interferometer. When the two photons, the key bit is this beam splitter, this 50-50 beam splitter in the top right-hand corner. When the two photons enter this beam splitter, and I measure coincidences at the output. So I've got two single photon detectors at the output, and I'm looking for coincidence, means I'm looking when I have a click on one detector, did I have a click on the other detector? And statistically speaking, you look at how many times that happens. So that's what a coincidence rate is. When I start changing, so in the quantum, if I just, one can do a relatively simple quantum calculation, which is sort of what the diagram is showing at the bottom here. When those photons are, so I have four different options for what the photons can do. They come into opposite ports. They can then enter, they can exit opposite ports, either by going straight or by being both reflected. Then I have the two options where the two photons come out bunched. So they both exit one of the two ports. So it's four options. When the photons are completely indistinguishable, the calculation shows that two of these probabilities, the two probabilities for exiting opposite ports, they cancel out and they disappear. So you're left with what is called a photon bunching effect, which is often referred to as Hong-Ou-Mandel from the three researchers who first realized this effect would happen in a paper published in PRL in 1981. So basically what happens is, how do I use this effect? As I change the delay in my interferometer on one of the two arms, I'm essentially introducing a degree of distinguishability between the two photons because they arrive at different times now. And you see the formation of what I'm showing you in the top right, which is this dip in the coincidence rate. When I have the exact same delay or arrival time between the two photons, they're completely indistinguishable. So I only see bunched photons. The photons either come out two from the top or two from the right-hand side of my exit ports. As I introduce a delay, I start to introduce distinguishability and I start to see coincidences appear again. So I have this Hong-Ou-Mandel interference dip sometimes referred to photon bunching. I will also sometimes use the terminology of G2 interference. Now, something that we will come back to later on is that a form of this bunching effect can appear also classically. So what I'm showing you in this bottom graph is, so if I were to just do a classical, send in a laser and do an interference measurement, the G1 interference is what gives you the standard interference fringes. But if I measure those interference fringes in G2, so I'm now looking for coincidences, I will see this blue oscillating line here. If I now put a phase dithering on one of the two arms in order to remove, when I average the G1 fringes, I will be left with this phase averaged red curve, which you can consider to be a classical version of the G2, the quantum Hong-Ou-Mandel or G2 dip. The quantum version is shown in the black curve, essentially the quantum advantage. So how is quantum better than classical in this case is that the dip is 2 times, the quantum dip is 2 times deeper than the classical dip. But I think the classical picture is useful, A, because it's easier to implement, but also gives an intuitive understanding, at least of where the part of the dip is coming from. So we've been working on this problem of, or this opportunity, let's say, of using this photon bunching technique for quite a few years now. Now, in the original paper by Hong-Ou-Mandel, they were proposing to use this dip to measure very precise delays in your interferometers. You can see in the top right-hand corner, as I change the delay, the dip disappears. And in the original paper, they are essentially proposing that, so the width of this dip is given by the inverse of the bandwidth of your photon. And typically this would be of the order of, the photon duration would be order of a few picoseconds, and the precision you can get by looking at how this dip shifts is of the order of a micron. And people had been, for the past 30 years, basically, been using this. So you scan the delay on one of the two arms, you measure the full Hong-Ou-Mandel dip, and you just look at where the position of the bottom, the peak, is in your dip to determine the path delays. And that gives you typically of order micron resolution. But actually, so what we looked at, we looked at this a bit more closely and realized that actually the bottom of the dip is the worst possible place to measure this these coincidence counts, because if one goes through sort of a Fisher information analysis, the Fisher information is proportional to a gradient of the parameter that you're trying to measure, that's the delay. So at this point here, you have an inversion point. That gradient is 0, meaning there's actually 0 or close to 0 information here. The best place at which to measure a change or a shift in that dip due to an object being placed in your interferometer is actually at the edge here, where the gradient is maximum. And if you do that, so basically what you do is you first do a scan where you measure the shape of the dip. That gives you a maximum likelihood estimator. And then you place the interferometer delay at the edge of the dip, and then you look for changes due to the introduction of a sample in the interferometer. You can show that you can get down to sensitivities of the order of a single nanometer, so, or attosecond time of flight delays in your setup. So this becomes fairly competitive to classical G1 interferometry. More recently, what we've done is we've tried to then extend this approach to the imaging domain, because up until recently, it had only been used as sort of like a single point sensing technique. And so what we're showing here on the left is basically how this works. So you have now a sample which has some form of spatially varying thickness. And what you look for then is the interference. You spatially resolve the Hong-Ou-Mandel interference at the output. And again, we're using this trick that I mentioned just a moment ago. You do a first scan, you look for an average dip position, you place the whole interferometer delay at this midpoint, that I'm showing with this dashed line here. And then you look for how, as across the image, you look for how the coincidence counts vary across the image compared to that point. And that will give you a one-to-one map onto the depth of the sample that you have in the interferometer. So this is what the setup looks like. We have a laser that's pumping a BBO crystal. I'm happy to answer also later on that question we had in the Q&As as to whether one could even use biological samples. The answer is yes. I mean, that has been done. And you don't need very high pump powers. I mean, you can use laser powers of order of a milliwatt and get photon pairs out. We then divide the two photon pairs into sort of the signal and idler are divided into the two arms. And we're placing the object over here, which is then imaged with a telescope onto the camera at the output. On the camera, we're imaging both outputs from the beam splitter so that we can see, we can detect correlations across the two images at the two outputs of the beam splitter. I wanted to add just a few quick words on how we actually do that. And there are different kinds of camera technologies that one can use to measure these correlations. So what I'm showing you here is just a comparison between, for example, an EMCCD camera, or between a SPAD. SPAD stands for single photon avalanche diode camera. And what I'm showing you is measurements of just the parametric down conversion. This is spontaneous emission from a crystal that will emerge in the form of a cone. This is just because of phase matching or momentum matching considerations. So basically what you do is on the camera, you fix a point, for example, this white cross, and then you look within the same image for a photon, for another photon that arrives at the same point. And you do that over many, many thousands or even millions of frames, and you can build up a statistically relevant sort of picture image of the correlations that you have between the white cross and all other pixels across to your camera. And you can see that because of the phase matching consideration, so in the parametric down conversion, phase matching will always lead to, in the far field, to anti-correlated photon pair emission. So that's why I see when I choose a pixel at this white cross, I will always see counts emerge at the anti-correlated position. Okay, and you have three different examples here. The nice thing about SPAD arrays is that you can gate them, and you can get, although they typically have fewer pixels, as you can see in the images here, but you can typically get extremely high signal to noise ratio. On top of that, you can also typically also have frame rates that are easily of order 100 kiloframes per second. So in other words, they can be at least 1,000 times faster than your EMCCD camera. So for building images like these, with an EMCCD camera, this might take several hours. With a SPAD camera, it can take several seconds. What we're actually doing in a bit more detail is we're building this joint probability distribution where I'm looking for the correlations within the same frame, but then I also need to subtract out what we call accidentals. So that is fake correlations that are due to detecting one photon and then the other count maybe being due to noise. And the way you do this is you calculate the correlations within the same frame and you subtract out the correlations across different frames, because we know that the correlated photons will appear at the same time. So any correlations happening across frames at different times are clearly spurious, they're due to noise. And so you basically just do this subtraction, but the sum here has to be done over many, many frames. So typically we're talking tens of millions of frames, okay? And that's why it becomes so important to have a camera which has a very high frame rate. So you can do it with an EMCCD camera, but it will take you hours. You can do it with a SPAD camera that will typically have lower resolution, but can take seconds. You can do this also. I was only showing you images in the far field or detection in the far field where photons will be anti-correlated. In the near field, they will be correlated. In other words, whenever I have a photon on one pixel, there's a high probability of finding it on the same pixel. So that's what I'm showing you in this left-hand picture here. There will always, however, with SPAD cameras at least, there will be a hole in the middle of this distribution, because a SPAD camera can only detect one photon at a time on the same pixel. So then what we typically do is then we fit this with a sort of a Gaussian to interpolate and pull out the full profile. Nevertheless, though, you can do, so what I'm showing you in this graph in the bottom left is the correlation. So remember, I've got the camera, I'm looking at the two outputs, and I'm looking for the G2 or the coincidences across those two outputs. And if I look at the anti-bunching, I get this dip. This is the Hong-Ou-Mandel dip that I was talking to you about earlier on. But I can also look at the coincidence. I can look at the probability now of finding two photons, either in the left-hand or the right-hand output of the beam splitter, and then I'll see a peak corresponding to the dip that I see in the anti-bunching case. A key feature that we had to pay attention to here was that in order to actually see a high visibility dip in the coincidences, one has to carefully mode match the size of the biphoton mode. Remember, I'm looking for two photons here. So if you look at these top graphs on the left, those show sort of the equivalent of a biphoton point spread function. We need to match that point spread function to the size of the pixel on the camera. If you don't do that, if you have too many, if the biphoton point spread function is too small, it means that I have lots of modes hitting the same pixel, and then you get it's very hard to distinguish photons that are coming from the same mode. Remember, we're imaging now, so we're multi-mode. You have different modes contributing to the same pixel, meaning that your sort of quantum interference will wash out. And the same also happens if the modes are spread out too far, then they're overlapping. You have multiple modes overlapping on the same pixels. So there's a sweet spot, which is what I'm showing you on the right-hand side figure here. There's a sweet spot where you tune the biphoton point spread function to be the same size as the pixel, and you get very high visibility. And we did this by simply changing the size of the pump diameter on the crystal. And so this is what the image looks like. It's just from there's no object placed in the interferometer right now. We're just looking at the parametric down conversion, spontaneous fluorescence. And you can see that as I change the delay, depending on whether I'm looking at the coincidences or the bunching, you can see as I move towards the delay, it becomes dark or very bright. And I then put an object in one of the two arms. If I just look at the single counts, just look at the photon counts, I see nothing. If I start looking at the coincidences, you can see there's an object here, but because of the very low resolution, just 32 by 32 pixels on the camera, it's actually hard to distinguish what is going on. So what we then did was just a 2x2 raster scan of the camera in the imaging plane. And now you can see, just by increasing that resolution by factor 4-fold, you can now very clearly see the sample, which is basically just the letters U of G, which had been etched into a piece of glass. So this is a perfectly transparent sample, and we're just looking at the change of depth, which is of the order of 8 microns in the sample. And then we can combine, for example, we started playing around with, remember, we've got the bunching and anti-bunching information. You can combine both of those images to get something which has lower noise signal. So all this was reported in a Nature Photonics last year. I now wanted to show you sort of some of the more recent work that we've been doing, where we've been applying this Hong-Ou-Mandel sensing concept to the problem of fluorescence lifetime imaging. So summarizing a couple of things, this quantum Hong-Ou-Mandel interference effect actually doesn't require pure quantum light. You can also obtain this quantum advantage with classical light in one arm, just a laser, for example, or thermal light, plus a single photon Fock state in the other arm. So people have done this even, for example, people have seen quantum Hong-Ou-Mandel interference by using a single photon from a quantum dot. The other light was coming from the sun. And of course, I've already mentioned that you can actually just do a pure classical Hong-Ou-Mandel sensing. So let's just bear that in mind as we go forward in the next slides. So what we have been trying to do is measure fluorescence lifetime using this Hong-Ou-Mandel approach. And so this is an outline of the setup. In the simple version, we don't actually use this DS detector. We just send light into the beam splitter. That can even just be a laser pulse. And we use another output from the laser to pump our sample, so to stimulate the fluorescence. And then we collect the fluorescence and we look at the interference between the fluorescence and the laser beam. But then what we're measuring is the coincidence counts between the fluorescence and our reference beam, which can either be a single photon or even just a laser. Now, the reason we're doing this is because, I mean, we could just detect the fluorescence lifetime using one of these detectors and doing standard time-correlated single photon counting. And if you do that, so just looking at detector D1, for example, if you do that, you are limited by the impulse response function of your time-correlated single photon counting device. And that will typically be of order 100 picoseconds. So if you're measuring lifetimes, which are in the nanosecond, 500 picoseconds upwards regime, that's fine. And that's sort of the golden standard at the moment. But if you have fluorophores that have a decay time which is of the order of picoseconds, then you will not be able to measure anything with that technique. But remember, what I was mentioning earlier on is that the Hong-Ou-Mandel or G2 resolution, which in this case would be the equivalent of our impulse response function, is given essentially by the inverse of the bandwidth of your photons or of your reference photons. And these days, 100 femtosecond pulsed oscillators are the norm. So in other words, what I'm saying is that now, by using this G2 interference technique to measure your fluorescence lifetime, your limit is basically the duration of your photons, and now we're talking 100 femtoseconds or shorter. So you're immediately 3 orders of magnitude better than your standard SPAD detector in terms of the temporal resolution. And you can also do this in the single photon regime. So basically this graph now is just summarizing that concept that the lifetime that you measure is sort of, very hand-wavingly, the convolution between your Hong-Ou-Mandel dip and your actual fluorescence lifetime, and you can get resolutions which are order 100 picoseconds. So here are some experimental examples. We've got 3 measurements here. I'm plotting this in log scale of 3 different molecules that have lifetimes in the range of order of a picosecond up to 10 picoseconds that would not be measurable with standard TCSPC photon counting techniques, but here are very clearly, with actually very good signal to noise ratio, very clearly resolvable using our technique. The measurements I'm showing you in the top left with this very high signal to noise ratio did take a few minutes acquisition. We did that purposely to get that high signal to noise ratio, but you can take the same measurements in just a few seconds and still get acceptable results, in the sense that they're giving us the same kind of, we're giving us the same numbers basically in terms of recovering the actual lifetime of these molecules. Now, one of these molecules, the first one, this 4-DASP molecule, in terms of applications, at least for us, is particularly interesting because this forms part of a family of molecules called rotors. So these rotor molecules, when they, upon optical excitation, they undergo a folding, and in doing so, they become very sensitive. Their rotational states become very sensitive to the surrounding local viscosity of the medium where they're placed. And so they are used to sense the sort of the nanoscale viscosity in biological samples. So basically what is happening is, as the viscosity is changing, you start to cut off some of those rotational states. So by cutting off those rotational states, Fermi's golden rule basically tells you that that leads to a change in the decay time from the excited state. And so we went ahead. So these have been used in the past to measure, and specifically also the 4-DASP molecule has been used in the past to detect changes in viscosity. It was used primarily as by just looking at the intensity, and so doing ratiometric measurements. But these ratiometric intensity measurements are very delicate and tricky. You need to have very precise calibration, and from sample to sample, that calibration can change. What you really want to measure is lifetime. The lifetime of 4-DASP is very sensitive, extremely sensitive. It changes by orders of magnitude when you change the viscosity. But up until now, people had only been able to use it in a very high viscous regime, for example, pure glycerol, because only then did you have lifetimes which were long enough to be measured by standard TCSPC. But now, because we have this new technique, we are able to measure viscosity, which is I'm showing you in this graph, which is all the way down in the single-digit regime. So 1 corresponds to pure water. And here we're just adding very small percentages of glycerol, and we can still, you can see we're measuring from 10 picoseconds up to above 100 picoseconds, but we can measure in this very low viscosity regime close to water, which is actually what you want in many biological samples. So previously this wasn't really it wasn't really that useful, and now it is. So for example, 4-DASP has been used before to, for example, it's been applied to the study of blood cells. So this figure on the left-hand side is just an intensity measurement, which was reported by a different group. This is not ours, by Briol et al. In this 2021 paper. What I'm showing you on the right-hand side is not fluorescent for the moment. This is just a passive Hong-Ou-Mandel image of blood cells, where you can clearly see the shape of the blood cells. Here we're just measuring the temporal delay of photons as they go through the blood cell. So this is just showing that you can use Hong-Ou-Mandel or photon bunching to measure the blood cells. And now what we want to do is now augment this right-hand image also with the fluorescence coming from these 4-DASP molecules. And so in the same image, or in the same setup, you can get refractive index profile of your cells, combined also with a viscosity measurement. So that brings me basically to the end of my talk. So summarizing, so this fluorescence, this FLHOM, fluorescence lifetime Hong-Ou-Mandel measurement technique has a resolution that does not depend on the sensor impulse response function, but only on the inverse of the photon bandwidth. So it can give you extremely high resolution. This enables nanorheology for measurements of cell and subcellular viscosity and stiffness. And we're also looking at applications in neurosciences, because brain tissue viscosity is directly linked to neurodegeneration. And I could give a whole different talk on that, maybe for another time, where we're using a slightly different technique at the moment based on diffuse correlation spectroscopy, where we have indeed been able to make a very clear link between how the properties of light propagating through neurodegenerated, this is mouse brain tissue, and neurodegeneration. Nanoscale viscosity also plays a role, for example, in cancer cell motility. And we've also got a project where we're working on the study of carbon intake in oceanic microbe colonies. So I'll stop speaking here and happy to answer any questions.
Okay, thank you very much, Professor Faccio, for your inspiring talk. There are 2 questions in the chat. First question